Hello, students. Thought I'd do a quick screencast to talk about last week's test and make sure that we go over the rubrics and things like that. Before I get into the specific rubrics, I just wanted to highlight a document just recently posted to the Schoology page. It's this one right here, FRQ Tips and Tricks. There's just some general things that are probably good to talk about, and we'll hit these more as we go through. But I'll let you click and kind of read through this on your own. That is well worth doing in preparation for the exam, since it is in fact just a short answer. But there's just a couple things I wanted to highlight right now that kind of came up on your test. The first thing is a point that we haven't really made maybe as big a deal of as we should have, and that is your essay should be incomplete sentences, not bullets. So yeah, I'll show you specifically where I was not seeing complete sentences. It says here AP readers are instructed to skip non-paragraph responses. So they're told don't read bullet points. And this document, by the way, was put together by a group of readers. So, you know, use a complete sentence, even if it seems a little unnecessary. It's worth just give it a subject and a verb and make it a sentence. The other thing is, if the essay is set up with internal parts, answer them that way in order and clearly label each part. So I just noticed a number of people didn't really clearly put A, here's my answer, B, here's my answer. I am told that if you answer part A under the part B label, that the reader will still catch that and give you the point. But there's no question, having myself graded many essays, when I see one that's A, here's my answer, B, here's my answer, it's way easier for me to find the answer that I'm looking for, and you want it to be easy for your grader to find your answer. So again, complete sentences, and make sure you break things apart into A, B, C as appropriate. And then, you know, there's a whole bunch of other stuff here that I really recommend you read on your own. So getting to our specific test questions here. The first one was these fossils of lobe-finned fishes, and you've got 3 samples of rock, 350 million, 370 million, 390 million years ago, and it says select the most appropriate sample to find that transitional species between lobe-finned fishes and amphibians. Well, the lobe-finned fishes are around 380 million years ago. Amphibians are 363 million years ago. So in between those two time periods is where you would see the transition. And so that is 370 million years. Notice on this rubric, there is a complete sentence. Rocks from 370 million years ago sample. So again, most of you just wrote 370, and that was it. So put it in a sentence just to be sure. I'm not sure how picky they would be on an answer like this, but just put it in a sentence to be safe. The justification then is transition fossils are between those two time periods that I just discussed. Part B, describe 2 pieces of evidence provided by fossils of a transitional species that would support the hypothesis that amphibians evolved from lobe-finned fishes. Descriptions include but are not limited to the following. So you've got a big old long list of characteristics that might be present on that transitional species. Note that we want to be as specific as possible. A few of you just wrote vestigial structures. Well, what vestigial structure? You know, be specific. And at the very minimum, define the term vestigial structure. But the rubric is calling for specific things. A lot of you talked about homologous structures. A lot of you talked about these characteristics, molecular evidence, that kind of thing. The second question was the math-based question. And where did I put it? Here it is. So there's actually 2 ways you could have approached this. It recommends that you use the graph. And notice that it says determine the frequency of the dominant allele. So if it's asking for an allele, that simply means big A. So not the homozygous dominant phenotype, but the dominant allele. So it's just looking for big A. In the population, if the frequency of homozygous recessive individuals is 0.75. So if we're talking about individuals, and individuals have 2 alleles, 0.75 is actually equal to Q squared. So you have to make sure, if we're asking for an allele, that's just big A or little a. If we're talking about frequency of individuals, that's going to be the big A, little a. or p squared, 2pq, q squared, etc. So you could either use the graph. So 0.75 is the frequency of the little a, and so I go on my graph and I scroll down to about 0.75, and it's right about there, and then I come on down, and then these top numbers represent the frequency of big A. So if I'm here, I come down and I'm at like 0.12, 0.13, somewhere in there. And it's kind of hard to do digitally, I recognize. The other way you could do it, I'm not sure it's going to work. Oh, can you see that? Can you see that? So you could just like say, forget that graph. I'm just going to do it mathematically. And if you do it mathematically, then you should see something like that. Notice that the accepted range on this was 0.12 to 0.17 because they recognize that when you're reading a graph, you might, be off a little bit, and that's okay. Note also that these math-based questions are no longer part of the AP exam, so you're not going to see something like this, but certainly the content is appropriate for this unit on evolution. The third question gave you this phylogenetic tree, and it said where where should that lysine mutation go? And it turns out that gorillas have that sort of mistakenly placed, that mutation of that amino acid, and only gorillas. And so that change must have happened after this common ancestor. A few people said it happened down here, but if it happened down here, chimpanzees and humans would also have that changed amino acid. Since only gorillas have it, it had to have happened right there. So that was 1 point for identifying that. I think it was position 2 on your test, the way we had to format that. The reasoning, gorillas are the only species with that amino acid. The mutation could only have occurred in the branch that uniquely leads to gorillas. If the mutation occurred on the main branch before the gorilla line, chimp and human lines would also be affected. Most people got that and did a good job. B, predict the most likely single mutation affecting amino acid 104 in the species and justify how beta hemoglobin can function normally in all 4 species despite the mutation. So some of you simply wrote missense mutation, and it's saying predict the most likely single mutation. It's asking you to be specific, and the specific mutation is a G to an A. And you figure that out by looking at the genetic code that's not in this rubric. But if you look at the arginine and look at lysine, they differ in the second codon. So a G to A mutation in the second position of the arginine codon. And you do need to say the codon, because you also could have said C to T in the DNA of the arginine codon, because the DNA would be C to a T. So you had to specify G to A in the mRNA or C to T in the DNA for that. And then finally, this is probably the most missed point, the justification of that. A lot of you said, well, this is a silent mutation because it's not affecting the individual. That is not correct. A silent mutation is one in which the amino acid does not change. And since the amino acid is changing from arginine to lysine, that makes this a missense mutation. So there is a change in that protein. And so the question is asking, well, why doesn't that cause a problem? And the answer is, this is one amino acid of many, many, many amino acids in this protein. And in this case, apparently, that single change is not enough to affect the functioning of this protein. And so, you could think about, it says here, there's a lot of amino acids with similar R groups. So maybe a polar amino acid is replaced with a slightly different polar amino acid. So that means the protein still folds just fine. It isn't a problem. Or maybe this particular change is not part of the active site for that protein, and so then it doesn't really affect its function all that much. So, so that's why a single mutation can happen without causing a big effect. Now, keep in mind, you have to look at it case by case, because sickle cell anemia is an example where, yes, one single mutation in one single amino acid is enough to make the difference between typical um, red blood cells and the sickle cell red blood cells. So a single amino acid change can cause a significant change, but oftentimes it doesn't, and it just has to be looked at on a case-by-case basis. So that's it for the rubric. If you guys have questions, we'll probably have to deal with those via office hour, email kinds of things. But hopefully you're satisfied with your test. Thanks.